from the lab introduction, you know that today we are going to be conducting gel electrophoresis. Gel electrophoresis is a technique that allows the separation of polar molecules based on their size. We're actually going to do 2 labs focused on gel electrophoresis. Normally, today's lab allows you to practice the techniques associated with electrophoresis, as some of them, like loading samples into the gel, are quite tricky. While you won't be doing gel electrophoresis yourselves today, hopefully this lab is still helpful at introducing you to the concepts associated with gel electrophoresis. I'll also be giving you some tips and tricks that will help you when you're hopefully able to do electrophoresis yourselves. Normally, electrophoresis is used to separate DNA fragments. However, today we're going to be using food coloring. This will give you a really great idea and visual of how the different colored pigments are separated out based on size through gel electrophoresis. The first thing I'm going to do is load the dye into the wells at the top of the gel. The wells are basically like small slits or pockets that will hold the sample until it is drawn out by an electrical current. To load the food coloring into such a small area, I'm going to be using a micropipette, which looks like this. So the micropipette allows for the transfer and measurement of very precise and small amounts of fluid. And basically it works through this button at the top. This button, if I twist it, will change the volume of solution that's drawn up into the pipette tips, which you see here. So by twisting the button one way or another, we can change the volume. So once we have our volume set, I'm going to set it at 10 microliters for right now. We are ready to start moving around food coloring. So to load the food coloring, I'm going to first push down the button, put the pipette into the sample of food coloring, and then release the button. This will draw food coloring up into the tip. Now that I have the right amount of food coloring in the tip of the pipette, I'm ready to load it into the well of the gel. To do this, I first put the tip of the pipette into the well, then push down the button. Then before releasing my thumb, I take the pipette out of the gel, and then I release my thumb. Remember, releasing the button sucks things up. So if we did that while the pipette was still in the gel, we just draw our sample right back up into the pipette. When you're done with each sample, you'll want to change your tip so you don't have any contamination. To do that, there is a sliding button here, and you simply push it down with your thumb and it will push the tip off your pipette. So in case that was a little confusing to you without seeing what that actually looks like, I am going to do a few practice rounds loading the food coloring into these small containers here. So I'm going to show you what that looks like, and hopefully while I'm doing that, you'll get an idea of how our pipette works. One quick note, whenever you use a pipette, you have to have a tip on it. The tip is designed to hold the sample or the fluid that you're working with. If the fluid gets into the actual pipette itself, the pipette will be damaged. So never use a pipette without a tip on it. The other thing I'll mention is you want to make sure you're holding your pipette the correct way. You'll notice there's a slight hook, and that hook goes over your index finger just like this. This gives you additional stability, as those wells that we're using with our gel are very, very small, so you want a good grip on your pipette. Okay, so as I mentioned, I'm going to transfer some of the food coloring out of these large containers and into our small tubes here, so it's ready to be loaded into the gel. This will also give you a better idea of what it looks like to use the pipette. And I'm actually going to change the volume on the pipette. I have it set to 30 microliters, and that'll just make sure that we have enough food coloring in each of our tubes to load the gel completely. So I'm going to get started, and I'll narrate what I'm doing as I do it. Okay, so starting off with our red food coloring, first I'm going to push down the button. I'm going to insert the tip of the pipette into the food coloring, and now I will release the button, and you see now I have our sample, which is our red food coloring, inside the tip of the pipette. So now I'm going to push the button down, which you see releases the food coloring, and take my tip out of the sample. And now we have just this very small amount of red food coloring at the bottom of our container, and that should actually be plenty for what we need. And like I said, we're also going to want to change tips between each sample. We don't want our red food coloring to mix with our green or blue or purple. So again, to do that, you're just going to push your thumb down on the sliding button, and that pushes the pipette tip right off, and you don't have to touch it. So I'm going to keep going and do the other 3 colors as well, so that you understand how to use a pipette. Again, for our green sample, I'm going to press the button down, put the tip of the pipette into the food coloring, release my thumb, and now we have a sample in the tip of our pipette. Now I'm going to eject it by pushing the button down. And now again we have a small amount of food coloring right at the bottom of our tube. Now I'm going to press this sliding button to eject my tip. So I'm going to do that 2 more times. I'll speed it up for you guys. The colors that we just separated out into our tubes are red, blue, green, and purple. And those will be the 4 colors we use in our gel electrophoresis demonstration. Each gel actually has 8 wells or pockets at the top, so that means that each color will occupy 2 different wells. Once the gel has been loaded, I will be subjecting it to an electrical current, and this will separate the pigments out based on their size. I'll be explaining more about that step once we get to it. So let's get started. So of course, I'm going to put my tip on, push the button down, put the tip into the sample, release the button, and now I have my sample. Now I'm going to very carefully find the first well, release the button, take the pipette out, and release my finger. And of course, we're going to change our tip, so using this sliding button, press it off, and we have loaded our first sample. So now I'm going to do every other color, just like I did for that first one, until all 8 of our wells are full of food coloring. Okay, so now our gel is fully loaded with the food coloring, and now we're ready to expose it to an electrical current. So I'm going to get that all set up, and then I'll explain kind of what's happening and how the electrical current works to separate the pigments while that runs. All right, so I have placed our gel into this container here. This is called a gel box, and you can see that it's connected by 2 wires to our power supply here. So this power supply is going to push electrical current into our gel box, and that electrical current is going to separate the pigments based on their size. So again, you might be wondering exactly how that works. Well, the reason we have 2 wires here is because we actually have a negatively charged current running through our black wire to the top of the gel, and a positively charged current running through our red wire to the bottom of the gel. Now, the food coloring samples we loaded are actually slightly negatively charged, as they're polar molecules, and so they're going to be attracted to the positive end of the gel, or the bottom of the gel. So that is going to pull them down towards that end of the gel and out of the wells. Now, basically, how this works as far as the separation according to size is you can think of the gel like a swimming pool filled with Jell-O. And think of the molecules as either an elephant if they're large, or a goldfish if they're small. So an elephant in a swimming pool full of Jell-O is going to have a pretty difficult time. It's going to face a lot of resistance and is probably going to get stuck pretty early on. Now, a goldfish, on the other hand, because it's so small, it will probably easily be able to move through this swimming pool full of Jell-O and may ultimately reach the other end of the pool. That's exactly what's happening with our molecules here. Our large molecules are getting stuck in the beginning of the gel, while the smaller ones can move further and they move to the end of the gel. So once we finish and the molecules have been separated out, the ones that got stuck at the top of the gel are the larger molecules, and the ones that move down to the bottom of the gel are the smaller molecules. So I'm going to turn on the electrical current here. Once I do, you should see bubbles rising out of our gel box, and I'm going to let it run for a little while so that we can see these colors being separated out from each other. The gel has now been exposed to electrical currents for 40 minutes, and we can see some separation and movement has occurred. So let's take a look at our results. So the first color that we used in both the first and the second well was red. Now you may know red is a primary color, which means the only pigment in red is red itself. So we didn't see any separation here. However, we can see that red moved pretty far down the gel, which means that the molecules making up the red pigment are fairly small. Then in wells 3 and 4, we had green. Now green is made up of two different colored pigments, blue and yellow, and you can see those were separated out. You can also see that the yellow pigment is slightly smaller than the blue pigment since it moved further through the gel. Now blue, which is our next color in wells 5 and 6, is also a primary color, so we do not see separation here, but again we can see that it moved down the gel. Now blue appears to be a slightly larger molecule as it did not move quite as far as the red or yellow pigments. And then lastly, in wells 7 and 8 we had purple. Purple is composed of red and blue, and we can see some of the red started to be separated out from the purple. It appears some of the red remains because we still see a mostly purple pigment, but if we would have let this demonstration run a little longer, we may have seen more red separated out and simply blue left behind. Again, it appears the red molecule is slightly smaller than the blue as the red moved further down the gel. So you're going to want to make sure to draw this on your handout. You can do this in many different ways, but make sure you reflect the colors. So you can use colored pencils, crayons, markers, you could even do it on your computer, but you want to show how the different pigments were separated from each other and how far they moved down the gel. One thing I haven't mentioned yet is the gel itself and how that is prepared. It's actually very similar to Jell-O. We use something called agar, which is a powder made from algae, and we add hot water to dissolve that. We then pour that mixture into a mold, let it set and harden for 30 minutes to an hour, and we also use a comb to make the wells. Basically, the comb leaves behind spaces where the sample will later be loaded into. And that's pretty much it. Really simple process to make the gel itself. So again, make sure your handout is complete, and as always, if you need help, ask your teacher.